Hi, my name is Alyssa Baker, and I am a master's student at Clemson University. Today I will be presenting on the complete mitochondrial genome of the brown spiny lobster Panulirus echinatus with insights into phylogenetic relationships in the Achelata. The infraorder Achelata includes the spiny, slipper, and coral lobster families. Members are characterized by motile phylosomal larvae, enlarged first antennae, and no chelae or claws. Within the family Panuliridae, the genus Panulirus inhabits shallow tropical and subtropical oceanic waters. Panulirus is the largest genus of spiny lobster, with 20 recognized species. One species, Panulirus echinatus, inhabits rocky reefs and beds of calcareous algae along the coast of Brazil and islands of the southern and eastern Atlantic Ocean. Currently, the economic value of the Panulirus echinatus fishery off the Brazilian coast is valued at over $60 million and rising with the reduced harvest frequency of the primary catch, Panulirus maripurpuratus. Our study aimed to assemble and annotate the first complete mitochondrial genome of the brown spiny lobster Panulirus echinatus. With increasing ecological and economic concerns surrounding this species, it is important to establish genomic resources for use in population studies, phylogenetic research, and the development of new management practices. First, muscle tissue samples were extracted from specimens collected in Brazil. Illumina paired end shotgun sequencing was then conducted and over 3 million reads produced. The mitochondrial genome was then assembled using the GET organelle pipeline. Next, we annotated and curated the genome for depiction and further analysis. Then, we analyzed nucleotide composition, codon usage, and selective pressures on protein coding genes. Additionally, we examined tRNA and controlled region structures. The complete Panulirus echinatus mitochondrial genome produced by this study is represented in Figure 1. Overall, the mitochondrial genome was 15,644 base pairs long. Most genes were recorded on the H or heavy strand. For protein coding, both rRNA and 8 tRNA genes were coded on the L or light strand. The AT codon bias was also weaker in Panulirus echinatus than in other known Achelata mitochondrial genomes. We also observed that protein coding genes primarily use conventional start and stop codons. Exceptions included the COX-1 initiation by the unconventional codon ACG, which frequently occurs in Panulirus species but is less common in other Achelata. Additionally, both COX-1 and NAD4 terminated in the incomplete stop codon T, which is a widespread occurrence in the genus Panulirus and family Panuliridae, and occurs to a lesser extent in the family Scalaridae. Selective pressure on protein coding genes is displayed in Figure 2. We found all protein coding genes underwent purifying or negative selection, where genetic diversity is reduced via the removal of alleles or deleterious mutations. This is consistent with Panulirus argus research, although selective pressure on protein coding genes is not extensively studied in Achelata nor in other Crustacea. Selection was strongest in COX-1, COX-2, and COB, and weakest in ATP8, NAD2, and NAD6. As depicted in Figure 3, tRNA followed standard cloverleaf structure. Although the serine 1 T-arm was reduced, there were no drastic changes such as those observed in other Panulirus species. tRNA also used anticodons TCT for serine 1 and TTT for lysine, which is generally uncommon for invertebrates, but is known to occur in other Achelata. rRNA exhibited higher AT composition than that of the total genome. Lastly, we observed a 733 base pair long non-coding control region with 7 repeating sequences but no tandem repeats. In conclusion, we performed the first assembly and annotation of the complete mitochondrial genome for Panulirus echinatus. Our next step for this project will be to conduct a selective pressure analysis on Achelata complete mitochondrial genomes. This knowledge will apply to future research into the ecology and phylogenetic relationships of species within Panulirus, the family Panuliridae, and the infraorder Achelata. Thank you for watching, and if you have any questions, please notify me in the chat room or in the comment section of this video.